to another video in my beginner's guide to Budgerigar Genetics. And in this video we're going to look at multiple alleles. So what do I mean by that? Well, we know that a gene can mutate, as we've seen in the blue mutation, where the gene that produces the yellow pigment in the bird mutated, stopped producing that, and therefore we end up with the green bird that looks blue. But what can happen then if a gene was to mutate more than once? And does this indeed happen? Well, clearly if a gene can mutate once, then it can mutate again, and that's what's happened in a number of genes, including the dilute gene and the blue gene. I'm going to focus in this video on the dilute gene because there is still some discussion around the mutated blue gene, so the gene that produces both the blue Budgerigar but also the yellow face and the golden face in a type 1 and type 2. And I need to go through that in a little bit more detail, and I'll cover that in the next video. But for this video we're going to look at the dilute gene, and that's the mutation that produces the type of bird that we can see up here in the top cage. A very old pet type bird that I've had, now trying to get out of the cage. But that is indeed, that's a dilute variety. So, not the best of birds clearly, because it's only a pet type, but it is a dilute variety. And that's what we're going to look at in this particular video. So what I'll do now is we'll go over back to the computer, and we'll look at the various mutations that the dilute gene has produced, including the clear wing and the grey wing. So let's go over and I'll meet you there in a minute.
Hello, and here we are at the computer to look at the multiple alleles, and in particular how their inheritance is passed on. As I said, the one that we're going to look at in a bit of detail initially is going to be the dilute gene. And the first thing I want to do is to talk about how the multiple alleles around the dilute gene interact before we then look at the Punnett square outcome predictions for them. So if I open up this Word document in here, we've got an explanation of how the multiple alleles around the dilute gene inherit. So there are 4 possible genomes for the dilute mutation. So there is in fact up the top here you can see, so we've got the normal wild type, so where the dilute gene hasn't mutated at all. We've got the dilute mutation, and then we've got the grey wing mutation and the clear wing mutation, and I've written down the side here the various abbreviations that we're going to use when we come to the Punnett square and for future discussion. So the inherited order around the dilute gene is that the wild type gene, or the wild type alleles, is dominant over all other mutations. So in the same way as the green is dominant over blue, so the wild type, the non-mutated reduction in colour that the dilute gene produces is dominant. So the wild type is dominant over all the other mutations. The dilute grey wing and the dilute clear wing mutations are of course recessive to the wild type. They're co-dominant over each other, and I'll explain what that means in terms of what the bird will look like in a little while. And they are both dominant over dilute. So as I say, the dilute grey wing and dilute clear wing are recessive to dilute recessive to the wild type gene. They're co-dominant over each other, and they are dominant over dilute, and therefore you can tell that the dilute mutation is recessive to all other mutations. So what does that mean in terms of our genotypes and our phenotypes? So we can clearly have 2 of the normal non-mutated gene, which is just the wild type Budgerigar. We can then have a Budgerigar that contains one of the wild type genes and one of the grey wing genes, in which case it will look like the wild type will be split for grey wing. We can have a Budgerigar that contains the wild type gene and the mutated clear wing gene, in which case it looks like the wild type but is split for clear wing. And the final version of that is the wild type gene split with the dilute mutated gene, in which case we'll have a normal looking bird that's split for dilute. So all those birds will look normal, but there's a possibility they could be split for either grey wing, clear wing or dilute. Okay, so the next one down is we'll look at the dilute grey wing. So we could have dilute grey wing split for dilute grey wing, or a dilute grey wing gene with a dilute grey wing gene, in which case it will look like a grey wing. It will clearly be a grey wing bird. So we could then have a grey wing mutation with a clear wing mutation, in which case I said it's co-dominant, so it will be a full-bodied colour grey wing, and I'll explain in terms of the body colour at the end of this section of what that one actually will look like. The final version is we could have a grey wing mutation with the dilute mutation, in which case it will look like a grey wing and be split for dilute. So in terms of the grey wing, then we could have a full grey wing with no split. We could have a grey wing split to dilute, or we can have the in-between grey wing and clear wing mutation. For the clear wing, then we can clearly have a clear wing mutation with a clear wing mutation, in which case we get the clear wing. We can have this opposite way round to the one that we had up here, where we've got a clear wing with mutation with a grey wing mutation, in which case again it will be a clear wing grey wing, which is in effect is a full body coloured grey wing. And then we could have the clear wing and a dilute mutation, in which case it will be a clear wing split for dilute. So it will look clear wing and it will be split for dilute. And the final version then is obviously the dilute mutation with the dilute mutation, in which case it will look like a dilute. So the interesting thing here is the only way that you can get a dilute is if it is split for nothing. So it contains both of the dilute genes, whereas the others, there's some various combinations as we've seen there. So that's the various possible genotypes, and that's the order in which they go. So clearly the non-mutated wild type gene is dominant over all. The grey wing and the clear wing genes are dominant over dilute, and they're co-dominant to each other. So that's the inheritance order. What I'll do now is quickly explain what I mean by the co-dominance of the clear wing and grey wing and what that will actually produce. So let's just consider for a moment then the grey wing and the clear wing, where a bird contains both of those mutations. As I said, the clear wing and grey wing mutation is co-dominant or has incomplete dominance over each other. So there is an in-between stage bird. It in fact creates a bird called a full-bodied grey wing. It has the wing markings reduced by 50%, just like the grey wing, but has the full body colour and the cheek patches of the clear wing. So I hope that explains the outcome that you will get from the clear wing grey wing. So it's incomplete dominance or co-dominance, and there's an in-between stage, a little bit like the single factor spangle. There is an in-between stage in terms of that bird. So let's have a little bit of a practice some pairings then using the Punnett square. So here we are with our first Punnett square that you should all recognise, and we're going to look at pairing two normal split birds. So the first one across the top here is a normal split clear wing, and the second bird is a normal split dilute. It doesn't matter which is the cock bird and which is the hen, as neither of the mutations are sex-linked. So let's have a look at what the possible outcome is. So the normal split clear wing can pass on either the non-mutated gene or the clear wing gene in terms of the two alleles, and the normal split dilute can again pass on its normal non-mutated gene and the mutated dilute gene in terms of its two alleles. So in this square here then, and if we bring those two across, we can see that it's going to carry over both of the two normal non-mutated alleles. In this square here, if we look at this one, it's going to inherit both the dilute clear wing gene or mutation and the non-mutated wild type alleles. The bottom square here, once again we're going to have the non-mutated wild type gene, and this time though it will also inherit one of the mutated dilute genes, and this one here will be a dilute clear wing mutation, and it also inherits the non-mutated, the mutated dilute gene. So what would that mean in terms of our expected outcomes then? So we'll probably see that we know that this one here is just going to be a normal type bird. So get this right. So there we go. That's just a normal type bird. The one across the top here, however, is going to be, we'd have to look at what the inheritance was. And if we remember from our inheritance that we looked at earlier, so this inheritance here, we'll remember that the wild type gene is dominant over all other types. So this particular bird here, the outcome here, will actually be a normal split clear wing. So down here, once again, we contain one of the non-mutated wild type genes, so we know that it's going to look, its phenotype is going to be normal, and this time it's going to be split for the dilute gene. So, and the final one here is slightly different, because in this case we've not got any of, it's not inheriting any of the non-mutated wild type genes. In fact, it's inherited one of the mutated clear wing genes and one of the mutated dilute genes. Once again, if we remember back here, we know that the clear wing gene is dominant over the dilute gene, because dilute is recessive to all other types of genes. So in this case here, we're going to actually have, in terms of that bird there, what we will actually end up with is a clear wing split dilute. So we will end up, in terms of our outcome predictions, 75% of the birds will look normal, of which 25% of them will be normal without any splits. 25% of those birds would be normal split for clear wing. 25% of the birds would be normal split dilute, and finally 25% of the birds will have a phenotype of clear wing, and a genotype will mean that they'll be split for dilute. We know that all the clear wings that produce from this pairing must be split for dilute. So there we go, there's the 25% there. So that's a fairly straightforward version of the Punnett square. Let's have a look at what happens if we take this a bit more further. We start to look at some crossings between the mutated type sort of clear wings and dilute type crossings. So what I should do is we'll move on to this Punnett square here, where we're looking at pairing up the clear wing dilute, split dilute, with a grey wing split dilute, and see what the outcome is of those particular pairings. So let's have a look at the outcomes of this particular pairing then. As I said, we've got a clear wing split dilute with a grey wing split dilute. So in this top square up here, the one we're looking at, it's going to inherit one of the mutated clear wing genes and one of the mutated grey wing genes. And for, if we remember our outcome for that, it means that we're going to have a full-bodied grey wing bird. In this one here, because they're co-dominant. In this one here, we're going to inherit one of the dilute genes and one of the grey wing genes. in this one here, we're going to inherit one of the clear wing and one of the dilute genes. And this outcome here is going to be inherit both of the dilute genes. So in terms of the predicted outcome then, we can see we've got a number of different types of phenotypes coming out and genotypes coming out from this particular pairing. So the first one here, the top square here then, we know that if you inherit one of the clear wing and one of the grey wing genes into a single bird, then there is incomplete dominance, and therefore that particular pairing is going to produce a clear wing, grey wing bird, which will in effect be a full-bodied colour, a grey wing. So, and that's going to represent 25% of our possible outcomes. This one here, where we've got a grey wing split dilute. Remember, dilute is recessive to all other gene types. So this bird here will in fact have a phenotype of grey wing, but it will actually be a grey wing, a split dilute. So 25% of the birds, once again, will be grey wing split dilute. So there'll be a grey wing phenotype. The one down here is very similar in that once again the dilute mutation is recessive to the clear wing mutation, so we're going to end up with a phenotype of bird that will look like a clear wing, but it will be split for dilute. So let's put that one in. So 25% of the birds will be clear wing split dilute. Final one here, we've got inheriting two of the dilute genes, so we know that the bird must therefore look like a dilute, so it's as a pheno and genotype are identical, and that it will be a dilute bird. So there we go, 25% of the birds will be dilute. So in terms of our possible pairings here, we're going to end up with I suppose 50% of the birds looking similar to the parent, so that 25% will be grey wing in terms of and split dilute, and 25% of the birds will be clear wing split dilute, so they'll be identical to the two parent birds. We're then going to end up with a grey wing that is full-bodied, something that neither of the two parent birds were, 25%, and 25% of the birds will be dilute in terms of what they will look like. So in terms of what the bird will be 25% of them will be dilute. So we're going to get from that pairing to 50% of the birds will not look like either of the two parents, and we'll actually end up with some dilutes. So interesting outcomes in terms of the pairings where we've got multiple alleles in a pair of birds. So hope you enjoyed that and hope that made sense. What we haven't covered in this video is the multiple mutations of the blue genes. That's because that particular mutation is a bit more complex, and I will try and go into that in the next video. There is still some debate about the various mutations of the blue genes, but once again that can wait till the next video. What we'll do now is go back to the birds and finish off there.
Well, once again, that's just about all we've got time for in this video. I hope you enjoyed it, found it interesting, and do play around with the various Punnett squares for that particular multiple alleles. And what I said at the beginning of this video, in the next video we'll try and look at the various mutations in terms of the blue mutation beyond just the standard blue. So the yellow face and the type 1 and type 2 and the golden face. But until then, please, if you did enjoy the video, give it a big like, and if you want to see more of these, then why not subscribe.